In this section, we're going to talk all about passive immunization, and another name for this is antibody therapy. So remember, with passive immunization, we're not creating our own immune response against the pathogen, but instead, this is a temporary protection with antibodies to help fight off a pathogen that may have entered the body, or if there's an exposure, it would be a way to treat the pathogen before, any disease actually happens. So, in a way, although typically we think of vaccines as more of a preventative rather than a treatment, a passive immunization is more of a treatment. It can actually be an alternative to antibiotics. So, if we don't have an antibiotic available for a certain pathogen, or if antibiotics are not enough, or in the case of viruses, we can't even use antibiotics and antivirals might not be the best, we can instead use some passive antibodies to help treat and prevent the infection from getting worse. Let's say that we have a bacteria that has invaded the body and we don't have an antibiotic for this pathogen, or this is a really, really bad pathogen and antibiotics aren't going to be enough. We could provide the person some antibodies, and these antibodies can do a number of things. One, if the antibody is bound to flagella, now the pathogen can't move. Also, remember that when antibodies are bound to an antigen, this recruits phagocytes to come in and engulf the pathogen. And remember, the name for this is opsonization. So having passive antibodies will help opsonize or help those phagocytes come in and eliminate the pathogen. Now, maybe this pathogen makes some toxins, all right? So it excretes some exotoxins that cause a lot of harm. Exotoxins are what actually damage the tissues. So again, there could be different antibodies that will come in and bind to the toxins. And now that the toxins can't bind to their target cells, this will neutralize them. So passive antibodies could also be used in neutralization against the toxin. Similarly, we could have a virus that infects the body, and we can use neutralizing antibodies that will bind to the spikes of the virus, and now the virus can't attack the human cell. So again, this is another type of neutralization. Also, remember, if a human cell is already infected with a virus, sometimes what can happen is a very sick human cell will no longer present any MHC or MHC2 molecules on its surface. And this will actually lead to antibodies that are going to bind all over the surface of this sick infected cell. And these antibodies are going to attract natural killer cells. And remember, natural killer cells will come in and release their death package and destroy this infected human cell. So passive antibodies serve a lot of functions in helping to treat and eliminate pathogens before the disease becomes too severe. There's four main ways that we produce and use passive immunization therapies. And I'm going to go through each of the four ways and give you some examples of when we might use this in real life. So the first thing we could do is we could take a healthy person who has been exposed to pathogens just like all of us have, and that healthy person can come in and they can donate their serum. They could donate blood, we can extract the serum, and from their serum, we will extract plasma, which has all sorts of random and good antibodies against all varieties of pathogens. So in this case, we're just using total human serum globulin. We're getting a wide variety of antibodies from this donor. Then we could take this plasma and we could give it to somebody who needs it, somebody who's very severely immunocompromised. So for example, this might be something that somebody with AIDS who's very immunocompromised, they might need that they might need a regular supply of antibodies to just keep them healthy. And there's many ways we might give these antibodies to that patient. We could give them intramuscularly, subcutaneous under the skin, or intravenous. So again, some examples may be a newborn baby who has a primary immunodeficiency disorder and they basically have no immune system, somebody with AIDS, somebody who's very immunodeficient or immunocompromised, maybe somebody who's just recently had a organ transplant and they just need those antibodies to help aid their immune system. That's one thing we could do. The next thing we could do is we could take a very high titered serum from a donor that has a high titer of a specific immunoglobulin that we're interested in. So for example, when COVID first emerged, there were people who got sick with COVID, they had very minor illness and they recovered quickly. And these people had those golden antibodies that we wanted and that we desired. So we could take that serum from the recovering patient and we could give that serum to somebody who has very severe disease, who was very sick in the hospital, and those antibodies might have saved their life. And we actually did do this a lot during the early days of COVID when nobody had any prior exposure and no antibodies to it. We could take antibodies from somebody who recovered very well. So in this case, they had a very high titer of those COVID specific antibodies because they had recently been infected. And when the more recent the infection, the higher the titer is going to be for antibodies against that pathogen. So this is similar to the first example, except in the first example, we're just taking a huge variety of whatever's there. We're not looking for anything specific. In the second example, we're looking for something specific based on a pathogen that somebody may have recently recovered from or been recently vaccinated against. So they're going to have a very high titer of antibodies to that specific pathogen. And let me give you another example of when we would use a high titered immunoglobulin from a human. If any of you have ever met me, you know that I am terrified of only one thing, and that is rabies. And rabies is a virus that is very slow acting. So somebody could be bitten by an animal or come in contact by animal scratch or close inhalation with the rabies virus. But the one good thing is that there's plenty of time between the time of getting the virus before it produces signs and symptoms. And the good thing about that long incubation period is plenty of time for us to use this passive immunotherapy to help prevent rabies. The thing about rabies is that once clinical signs and symptoms begin, such as in the early phase, we see fever and vomiting and headache. And then once it progresses further to like coma, there's no possibility of treating it at that point. So that initial time period, that initial incubation period is really important for preventing rabies before it gets to these signs and symptoms. And this goes for animals as well as humans. So with rabies, this is the most serious virus because it's 100% lethal if somebody contracts it and doesn't get treatment. We use a variety of different types of vaccines to help prevent and treat it. One of these is a passive vaccine. And what we do is some people who are volunteers or even veterinarians or people who work closely with animals that potentially could have rabies, they actually get vaccinated using a preventative vaccine where they're going to build their own immune response and their own antibodies against the virus. And then what we can do is we can take the serum from those people that have been vaccinated and have a lot of good antibodies against rabies, and we use those antibodies as a passive vaccine. So if somebody is bitten by a rabid animal, they'll go get a huge amount of doses of this human rabies immunoglobulin, which has a lot of those antibodies that we want to help prevent the rabies from progressing and actually becoming serious disease. We'll also give them an inactivated rabies vaccine just as a precaution. Why not give them both? And now these people have been vaccinated against rabies over a period of time. They'll start making their own antibodies against the virus. We can use their serum that has all those good antibodies that we want against rabies as a future passive vaccine in case someone else is exposed down the line. Another example of where we use a passive vaccine from a high-titered human donor is anthrax. In 2001, anthrax was actually used as a bioweapon. Somebody, actually a U.S. American scientist, purposely planted anthrax bacteria into letters in the mail and sent it to a bunch of senators and to a bunch of news media people. And there were actually several people who contracted anthrax and died because anthrax produces a very potent and deadly toxin that kills people. So what we do in the event that somebody is exposed to anthrax is that actually some military recruits or volunteers or people who maybe work in a lab where they're working with anthrax and they have potential of being exposed to it, we actually vaccinate them against anthrax. Now, we don't just give everybody an anthrax vaccine because it's such a rare disease. It's not worth the risk of even working with it to make the vaccine. So those military volunteers that are willing to get the vaccine, now they're going to make antibodies against anthrax, and we can take the serum from them and we can make anthrax antitoxin from human sources. And we actually have enough anthrax antitoxin stockpiled for everybody in the United States, just in case there's ever another event like this or worse. So these are all cases where we're taking antibodies from human donors and using them to prevent or even treat, if early enough, a pathogen. Now, 9 times out of 10, we're not going to take serum from human donors just because that's a lot of work. Getting the serum from the humans, getting humans to volunteer to give their serum, testing the serum and making sure the person doesn't have any infections that we didn't account for. So it's much easier to do the bottom two, where we take antibodies from animal serum or from animal sources. So the next thing we could do is we could get a wide variety of antibodies against a pathogen or antitoxins, so that would be antibodies against a toxin from an animal source. So here's how we do this. The first thing we're going to do is we're going to take an animal, and a lot of times we use rabbits, but we can also use goats or guinea pigs. So what we do is we take an antigen that we've purified. We usually don't inoculate the animal with a live, dangerous pathogen because, again, this all should be as humane as possible. But instead, we'll take like a part of the pathogen. So maybe we're just going to take, the pili or certain proteins from the pathogen. So the same way we would create a vaccine for a human, we'll take that antigen, instead of putting it into a human, we'll inoculate it into the animal. And now what's going to happen is that animal is going to have an immune response, just like a human immune response. Animals' immune systems work the same way. So their B cells are going to have specific receptors that bind to the antigen. The B cells become activated. Some of them form memory B cells. Some of them differentiate into plasma cells. And those plasma cells will make antibodies. And that's what we want. We want a diversity of antibodies against an antigen. And now we can take those amazing antibodies from the animal and we can use them as a passive vaccine for us humans. So why we would do this, again, is one, we're not using humans. We're not experimenting on humans and putting antigens into humans. Instead, we're using animals. So we can do this faster. We can make a lot more of the product we want. And again, because we're not looking for anything specific, we're getting a wide diversity of antibodies against the antigen. And the more different types of antibodies we have, that's usually better, because now if somebody had an infection and maybe one antibody wasn't as specific as we want, we have a lot of them. So hopefully, at least one of them will do the trick and get rid of the pathogen before the person gets serious disease. So let me give you some examples of where we'll use these polyclonal. By the way, polyclonal meaning we're getting many different types of antibodies, not just one single type. We're getting a nice diversity. So let me give you some examples of when we would use this. Recently, we had an Ebola outbreak as recent as 2016, 2017, 2018. One of the things that happened during the outbreak was there were some laboratory accidents, which happen all the time, believe it or not. And there was a nurse who accidentally stuck herself with a needle that had been in contact with a patient who had Ebola. And there was also a group of laboratory scientists who came in contact with active Ebola virus. So all of this medical team decided to take their chance on trying a passive vaccine against Ebola. And what they did was they actually did this process that I just showed you here, only they used a goat. So they took a goat and they inoculated it with some Ebola antigen, and the goat made a bunch of antibodies. And the medical team actually chose to experiment on themselves, and they inoculated themselves with the goat antibodies, the goat hyperimmune serum. And luckily, all of these medical professionals survived, and none of them ever showed any signs or symptoms of Ebola. So this was kind of a cool story. And I will tell you that I can't find any report of this serum ever been tested again because luckily we haven't had super significant Ebola outbreak like we did about 10 years ago. Another case where we use animal serum is for a pathogen called botulism. So you might have heard of botulism because it's often associated with canned foods. Like if you do your own canning at home, there's a risk of botulism because if the canned food is not properly preserved, there could be some bacterial spores that get in, and the bacteria will grow in the canned food, and this could also be dented cans. And this particular bacteria releases a very potent neurotoxin. It's called the botulinum toxin. And what this toxin does is, if you remember back to anatomy and physiology, at our neuromuscular junction, so the space between the neuron and the muscle, there's this neurotransmitter that's released, and the neurotransmitter is called acetylcholine. And this is the neurotransmitter that allows your muscle to contract. This particular toxin, the botulinum toxin, blocks the release of acetylcholine. So the muscles no longer contract if somebody's been poisoned with the botulinum toxin. So if somebody is exposed to this toxin, like say we knew of a food that had a lot of this toxin in it and we knew other people had eaten this food, we can quickly help prevent serious disease by using the botulism antitoxin. And we make this antitoxin in horses. So again, what we do is we inoculate the horse with botulism antigens. We take the horse serum, and then we use that serum as a treatment to help prevent serious botulism disease. Now, as you can imagine, there are some risks to using horse serum, and the biggest one is that some people may be allergic to horse or animal serum. And especially we wouldn't use horse serum on a newborn baby because that could also lead to anaphylactic shock, and it'd be very difficult to treat. And unfortunately, the number one people at risk for botulism are babies because of raw honey. Raw honey has a lot of bacteria in it, in particular, a lot of Clostridium botulinum, because Clostridium botulinum is normally found in the soil and in plants, and bees come in contact with it. So honey has a lot of this neurotoxin and a lot of this bacteria. Now, if an adult eats raw honey, they're usually going to be okay because we have normal flora. We have enough good bacteria in our body that it's going to outcompete this bad Clostridium botulinum. But babies, newborn babies, they don't have an established normal flora yet. So ingesting raw honey is very dangerous for babies because now the Clostridium botulinum can take over, it can release this botulinum toxin, and it can cause botulism in babies. So infant botulism is the number one type of botulism in the United States, and infants with botulism are typically on respiratory support and feeding tubes for about 6 to 9 months. So as you can imagine, this is no joke, and this is why they tell new parents that you can't feed babies raw honey. So in the case of babies, we can't use the horse immunoglobulins because it's not safe. So instead, we use a human immunoglobulin, like we talked about before. An example where we're inoculating a human with botulism antigen, we're taking the human serum and we're using that human sera for treatment of the babies. And by the way, the name of this treatment is called Baby Big, and that stands for Baby Botulism Immunoglobulin. Now, the big thing about this is it's extraordinarily expensive. It costs about $45,000 per vial for Baby Big. And as you can tell, that's why we typically use the horse treatment because it's going to be a lot cheaper than using the human serum. All right, the final type of passive immunization is instead of using animal sera with a wide diversity of different polyclonal antibodies, we can use them to create a monoclonal antibody. So just like it sounds, mono means one. So it means we're making one very specific type of antibody, just one antibody that's going to bind to one specific epitope. So for example, you've probably seen throughout your life, maybe on commercials, or maybe you or a relative have been on a drug like this that has this crazy long name that you can't pronounce, but usually ends in MAB. Whenever you see a drug like this that ends in MAB, that stands for monoclonal antibody. So there's, tons of these kind of drugs out there. And the way these drugs work are they are all very specific one type of antibody that's going to bind to one specific type of antigen. So for example, there are a number of cancer drugs out there that are monoclonal antibodies. If somebody has an autoimmune disease, we can often use a monoclonal antibody treatment. In this particular picture, it's showing monoclonal antibodies that were used for COVID in the early days before there were COVID vaccines available, and there's still some that are used today. And as you know, the best type of antibodies, the ones that are most specific, most desirable, are usually going to be IgG antibodies. So a lot of these monoclonal antibodies are IgG class specific antibodies to specific antigens. So let me tell you how we produce monoclonal antibodies. The process starts out similar to polyclonal, where we take an animal and we inject it with an antigen that we're interested in making antibodies against. We then extract the spleen from that animal. Once we get the spleen out, we'll homogenize it so that we get individual cells. And don't forget, the spleen has a lot of B cells, a lot of immune cells, but we're really interested in the B cells that are going to be the ones that are making antibodies. In a separate tube, we're going to make a culture of myeloma cells. Now, myeloma, this is a cancer cell line. So the good thing about these cells is that they can just grow forever. We can just keep proliferating them as long as we need to. But also, they're not going to be B cells in there. So these guys are not making antibodies of their own. They're just forever living cells. So now we're going to merge these spleen cells that are hopefully making our antibodies with the myeloma cells. And when we merge them together, we're going to create hybrid cells, which means they have a mixture of characteristics of the spleen cell where they're going to make antibodies and the myeloma cell where they're just going to proliferate forever. And from there, we can use a selective media and we can screen for the different antibodies that are made by the different hybrids. So one hybrid cell might make these green antibodies, one might make the red, one might make the yellow, but maybe we're only interested in the yellow ones because those are the very specific antibody that works best against the antigen. So then we can just use these ones for future experiments or for treatment against certain pathogens or cancers or whatever. So overall, to summarize, most of the time we're going to be making passive vaccines using animal source in some way because it's simpler and we can make more of it. The choice comes down to whether we want polyclonal antibodies or monoclonal. The biggest advantage of polyclonal is we're going to be able to make a lot of antibodies, a broad diversity that can target multiple different antigens. In the last week, we learned that there are certain antigens that might be similar amongst different pathogens. So with polyclonal antibodies, it might work against multiple strains or multiple species of that pathogen. Versus with monoclonal, we're only going to make one very specific antibody. But this might be a good thing. We might want to target just one specific antigen, and we might not want to have the potential for cross reactivity. So, for example, if we had a cancer cell that was presenting a very specific antigen associated with that cancer, we would want a monoclonal antibody because it would target that one specific antigen. So there's pros and cons to both, and it would depend on the situation of when we would use each. In this week's paper, we're going to read a paper about a pathogen called Clostridium difficile, or C. diff. And C. diff is another type of Clostridium like botulism we talked about earlier. But what C. diff does is it gets into the colon and it releases toxins that damage the intestines, and they cause really serious diarrhea that can actually kill somebody. Antibiotics are not effective against C. diff because C. diff makes endospores that can survive in the presence of antibiotics. So treating somebody with antibiotics can actually just make C. diff reoccur and make it worse. The two toxins that C. diff makes, the CDI toxins, are called TCDA and TCDB. And these are the toxins that lead to the inflammation that can cause something called toxic megacolon, which is really serious and can basically lead to toxic shock syndrome, organ failure, and death in severe cases. So in the paper we're going to read about a new strategy to help serve as a treatment for C. diff, since like I said before, antibiotics are not the best type of treatment for C. diff. And if any of you ever worked in a hospital or worked with patients, you probably heard of C. diff because it's a nosocomial infection, which means it spreads pretty rapidly in the hospital. And this is one of those high priority pathogens that we're desperately looking for more treatments and preventatives for.